Well, I can't tell. I don't know whether you can hear me either, but welcome back to Genetics Colloquium. We have another exciting week here with two very different talks this time. Our first speaker will be Ziyao Zhang from Cheng's lab, and I hope Cheng is there to do an introduction.
Oh, you don't have your camera. Okay, now can you see me, Nicole?
I can.
Okay, it's great. There's reverb, so maybe I should. So what up here, do I need to use this, that, or is that? I just shout? Okay, well, just reverb from there. Okay, well, thank you everyone for coming. I can't see everyone because of the light, and I know there are many of you on the Zoom. It's a pleasure for me to introduce today's first speaker, Ziyao Zhang, a third-year graduate student in my lab. I'll make this quick. Ziyao and I share something in common. We graduated from the same high school, many years apart, obviously. But we're here together, trying to do some interesting research. Ziyao graduated from the Fudan University, one of the premier undergraduate institutions in China. And she joined my lab about 3 years ago. And she's been working really hard and has collected a lot of interesting data. This is, she's fearless in doing all kinds of different experiments. And what she's trying to do in the realm of genetics, we're always interested in genotype-phenotype relationships, and she's trying to find something important to do, within the framework of the lab. And to find something important, she has been looking for really interesting phenotypes. And we use mouse models in the lab, and she has been analyzing a lot of mice and trying to look for something striking that is a result of certain genetic manipulation. And her reasoning is that if you find a strong and robust phenotype, then it would imply a very important function of certain genes and biological processes. And that process is long because mice have a longer gestation time and a longer lifespan than some of the other model organisms you guys are more familiar with. So it's taking her the long road to get to this point, but hopefully she's going to share some results today that indicate that there are interesting phenotypes in some of these mice she has been characterizing, and that would provide a strong foundation for the next step of the research, which is to figure out potential mechanisms. I don't know exactly which project she's going to talk about, because as graduate student, you'll know that you do many different projects in the lab. Even when they have the same title, they may mean different things. So very happy Ziyao is in my lab, and I feel very lucky to have her. And hopefully this is going to be the start of a very successful PhD training career. So without further ado, Ziyao.
I'll try to speak as loud as I can, and hopefully everyone can hear me okay. So my name is Ziyao Zhang. My topic today is the role of PrEP signaling in Rett syndrome pathogenesis. First, let us talk about Rett syndrome. Rett syndrome, or short as RTT, it is a neurodevelopmental disease primarily caused by loss of function mutation in MECP2 gene. And since it is located on X chromosome, it mostly affects females, and it affects about 1 in 10 to 15,000 girls in the US. RTT patients, they exhibit grossly normal early development, followed by a rapid psychomotor regression, losing their abilities of speech and purposeful hand use. And they may develop comorbidities such as seizures, autism, stereotypic hand movements, and so on. In mouse models, in our lab, we use mouse models. The loss of function or dysfunction of the MECP2 protein can lead to deficits in multiple aspects, including a range of behavioral changes in locomotion, emotion, sociability, and cognition. And they will have altered neural network excitability and deficits in synaptic plasticity and the dysregulated neuronal proliferation and differentiation. And sadly, there's no valid cure for Rett syndrome. The second hero in our topic is Krabs signaling. It is a very famous transcription factor and is expressed in a wide range of tissue and cell types and involved in a myriad of biological processes, especially in the development and function of central nervous system. The transcription activity of Krabs is believed to depend on the phosphorylation at serine 133 sites. There's not much work done about the connection between Krabs signaling and MECP2, but it has been reported that Krabs and MECP2, they can bind together and co-regulate some transcriptional activation. And in some circumstances, Krabs can regulate MECP2 expression through microRNA 132. But there has been no evidence suggesting how Krabs is involved in the disease, in the Rett syndrome. Okay. In 2017, our lab published a work found that both total Krabs and phosphorylated Krabs, they show reduced level in the forebrain neurons differentiated from MECP2 mutant cell lines. We tested 3 iPSC cell lines and 1 HCIC cell lines and saw the same trend. And that makes us wonder whether this altered Krabs level is involved in the RTT pathogenesis. In that paper, we did lots of cellular work. So we looked at several cellular hallmarks that are believed to be related with RTT pathogenesis, such as the reduced dendritic length. We did a transient transfection of Krabs plasmid, and by enhancing the Krabs expression, we saw there's an increase in the dendritic length and increased dendritic complexity and increased mitochondrial health. So basically, if we transfect with Krabs, there's seems to be an elevation, seems to be a rescued phenotypes in the RTT neurons. And after that, we used a pharmacological activation of Krabs with a drug called roloprem, which is a anti-depression drug. It can selectively inhibit the phosphodiesterase 4, which leads to reduced cycling AMT degradation and thus increase the pKa activity. And that will result in elevated Krabs phosphorylation. So with this, we did the same cellular analysis, and we saw first we did Western, and we saw that the roloprem treatment can induce pKrab level. And in the lower figures, we applied the drug to the MECP2 knockout neurons and treated with the roloprem. And the defects in the dendritic growth and the mitochondrial health were successfully rescued. And finally, we applied roloprem on the RTT mouse models. we use the female MECP2 heterozygous mouse. So they have been, so they received the roliprane, and we did some behavioral analysis. So first panel is the open field, which is a very classic assay to test their anxiety level. And from this one, this is the total travel distance. We can see the roliprane didn't show much change in the locomotion ability, but the roliprane treated mice, they spend a lot more time in the central area, suggesting that they are less anxious comparing to the ones who only received vehicles. And also we did a fear conditioning test. So basically this is a learning memory test. We give the animal some cues followed by electric shock, and 24 hours later we play the same cue and see if the mouse can react to the cue. So it's like if they can. Oh. What should I do? Okay, so should I continue?
Yeah. I just
Thank you. So yeah, and we used roliprane to treat the RTT mice, and we saw there's a reduced anxiety in the RTT mice, and also by doing the fear conditioning test, we see the mice that can memorize the cue better, suggesting they have a rescued memory, long-term memory. So basically that is our previous paper, and in that one we proved that pharmacological activation of crab alleviate RTT-related phenotypes, both in vitro and in vivo. However, it is still too early to conclude that crab signaling is the potential therapeutic target we are looking for, because as we all know, the drug activation, it is indirect and it may have some potential side effect. To further prove our hypothesis, that leads to my current project, which is we want to use a precise genetic manipulation to further validate the function of PrEP signaling in alleviating RTT phenotypes, and to understand the mechanism underlying. And that is this is the goal of my current project. And to do this, we employed a PrEP hypophosphorylation mouse line to achieve stable and precise PrEP activation, and to cross this with RTT mouse model. By studying the double mutant mouse line, we're hoping to prove that the enhanced PrEP signaling can rescue RTT-related phenotypes and can be used as a potential therapeutic target for RTT treatment. And yeah, next I want to show you the mouse line involved in this study. So first is the nestin-cree-induced MECP2 conditional knockout. The reason we use a conditional knockout is that a complete MECP2 knockout mice is very difficult to breed. And even with this conditional knockout, I have lots of difficulties. I had some troubles in the breedings. But anyway, with this conditional knockout, we want to, so MECP2 gene will be restricted. The deletion of the MECP2 gene will be restricted to the central nervous system under the control of a nestin-cree gene. And it will remove the third exon of the MECP2 and cause frame shift mutation. So basically, total loss of function. And the crab hypophosphorylated mouse model was generated by Dr. Rendell Tibet's lab here at UW-Madison. The mutation locates at the BS2 domain of the crab gene. So this is the crab gene. And the BS2 domain is responsible for the B56 binding. By disrupting this locus, it will inhibit the crab dephosphorylation via the B56 PP2A pathway, and in turn lead to enhanced S133 phosphorylation. So first, I want to validate the protein expression in the crab single mutant animal. And I did Western blot analysis from the adult crab homomutant mouse brains, and we confirmed that there's an increase in the baseline level of the P crab with no obvious change in the total crab level in all brain regions, comparing to the wild-type littermates. And from the bar graph, it shows the quantification of the P crab versus total crab level in the whole brain. And there's about 3 to 4-fold increase in the homozygous mutant comparing to the wild type. And here's my breeding strategy to generate the double mutant animal, and all the possible genotype combinations and frequencies of the male offsprings are listed below. Since the MECP2 gene it locates on the X chromosome, the heterozygous female mice will be mosaic for expression of either the wild-type MECP2 or the MECP2-KO allele of all their somatic cells due to the random X chromosome inactivation. So, and because in our project we want to see the phenotypes like faster and more obvious. So in that case, the hemizygous male mice, they are preferred for this for our current stage at least. And as you can see, because I have three alleles involved in this breeding, the frequency of producing a wanted genotype is very low. So that's one thing I said about one of the difficulty I came across with the breeding. So I'm still at the stage of accumulating animals, but I have accumulated some preliminary data, and I'm going to show my data in later slides. And the first thing we did after getting the wanted genotype is to confirm the protein expression phenotype. So this one, we're looking at the MECP2 expression. The protein were extracted from different breed regions of mice of different genotypes. From the Western blot, we can see the MECP2 level in the Tulox mice is actually lower comparing to the true wild type. And that is actually known to the field that the Tulox in the MECP2 may cause some decrease in the expression. And that actually told us I need to include a true wild type for all my later experiments. And also, when we look at the MECP2 conditional knockout, we can still see some signals in the brain regions. And that is actually kind of unexpected because we kind of expect the knockout to be cleaner. So that is another thing I need to keep in mind, just need to be aware the incomplete knockout may of this incomplete knockout, and I need to be more cautious with all the interpretation of the data. And this is the immunostaining data, and basically this is the same as what we saw in the Western blot. I used 2 different MECP2 antibodies to further confirm the results, and we can see in the Tulox again there's a decrease, but still lots of expression, and in the conditional knockout, most of the MECP2 is gone, but still we can see some signals. So consistent with the Western blot. Despite the incomplete MECP2 knockout, the mice, they're actually able to develop RTT-related symptoms. So here's a piece of video. Let's see if I can. Yeah, you can see this conditional knockout mouse, and you can see the hind leg clasping, and it's not moving a lot, and it's constantly tremoring. And when it walks, the hind leg is spreading wide, and actually it's not actually walking, but like bunny hop. And the coat is not very smooth. So these are all the phenotypes we're expected to see in the RTT mice. So yeah, since it can develop all these symptoms, we decide to continue using this model to study with this double mutant model. And the MECP2 Tulox mice, they appear to be normal. Okay. And after the MECP2, the next protein target we're looking for is the crab and the phospho crab. We can see the upper graph shows the total crab level in the different mouse lines, and we can see in the MECP2 CKO there's a decrease in the total crab level, which is consistent with our previous finding. And when we look at the phosphorylation crab level, we can see there's a very obvious increase of the phospho crab in the crab homomutant comparing to the crab wild type. So basically the model is working. And here's the immunostaining results again to confirm the expression we saw in the Western. So I have the previous was cortex, and this is the hippocampus. And we also there's some expression, some crab expression in the wild type, and we have more and more expression as we have the crab transgene accumulate. Since I'm still at a stage of accumulating mice, so right now I'm doing some basic observation and reporting. So first thing is the disease progression. To determine the disease progression, the male mice were scored weekly for a number of symptoms arising from the MECP2 deficiency. So each of these six phenotypes were scored from 0 as absent or as wild type, 1 as symptom present, or 2 as symptom severe. So the mice, they start to show symptoms at about 7 weeks old, and health conditions of the CKO mice, they get worse as they age and kind of plateau at the age of at 10 to 11 weeks old with some minor ups and downs. And it seems like the disease progression is, and it seems like the crab allele has little effects on the disease score in the CKO mice. However, something we want to be aware is in the MECP2 complete knockout mouse line, the disease score, they usually do not plateau. The mice, they just become sicker and sicker and eventually die. So this plateau curve we see in the CKO mouse line, that may mask some effects of the crab allele. So a complete knockout line is still needed to determine the effect of the crab signaling in the RTT disease progression. And another thing I did is just survival curve, because we want to know whether it can help prolong the lifespan of the RTT mice. And as you can see, right now I have only a very small sample size, and actually this is not a good enough sample size for survival curve. But from this we can see it doesn't seem like there's a very strong effect of the crab on the survival curve. But again, this is a conditional knockout, and a complete knockout line is still needed. And then so while there was little difference in the disease progression and the median lifespan, one observation caught my eye is that I noticed that there are many dermatitis and open wound observed in the double mutant breeding, and they are all happen in the CKO mice, as we can see here. And we tried several things like separating the animals, but didn't help, suggesting it's not due to the cage mate fighting, but they are actually self-barbering. They are pulling out their own hair. And as we can see from the figures, although I have small sample size, but it looks like the crab homo mice, they show milder symptoms and they have a later phenotype onset comparing to the crab heterozygous and ME2CKO mice. So that makes me wonder what this phenotype can be used as, whether this phenotype, what this phenotype indicates, and whether this can be used as an indicator, and how this could be quantified. Oops. So I look into literatures to try to find what self-barbering means. And this is usually linked with excessive self-grooming, so meaning the behavior of mouse cleaning their own body. And this is usually considered as a repetitive behavior, and it's usually linked with, it is usually used for a model for human obsessive compulsive disorder, or OCD. And although there are some debates, but lots of people believe it is linked with anxiety. And here's a previous studies I found. It reported that excessive grooming behavior was observed in the ME2 conditional knockout mice. So I try to do the same thing in my mouse line, in my double mutant mouse line. So basically what I did is I put each mouse individually placed in a clean cage for 30 minutes, and I have a camera to record everything the mouse does during the in the cage, and the time spent grooming is manually quantified. And over here is my quantification results. So we and we can see this gray bar, which is the crab wild type and ME2 conditional knockout mice. They spent more time grooming comparing to the crab wild type and two-lox mice, which I use as the control. So suggesting this is a strong phenotype. And the dark gray, which is the crab heterozygous, and this light gray is the crab homozygous mutant. And we can see there's a trend of decreased grooming time in the crab mutant mice. And so it's like there's a clear reduction in the self-grooming time comparing to the crab wild type and ME2CKO mice. Although the phenotype is not fully rescued in the 30 minutes duration quantification, but again, this is the number I show here. It's a very small sample size for behavior test, and I'm still keep accumulating data. And also, I believe this is an encouraging piece of preliminary results. I will, of course, keep looking at other repetitive and anxiety-related tests to try to confirm this. And another thing I try to look at, so this is something I did in the crab single mutant animal, is the fear conditioning test. First, we put the animal in a cage, we play a sound, we let the animal familiarize with the cage, play a sound, and then give the animal an electric shock. Then we wait a certain amount of time, place the animal back in the same cage to see if the same context can remind the animal of the fear memory. And after 2 hours after context test, we place the animal in a different cage. We change the contextual settings and play the same sound, and see how the sound can remind the animal of the fear memory. So this is the process of the fear conditioning test. I did 2 different tests, which is a 24-hour early long-term memory and 2 weeks late long-term memory test. And we can see in the crab single mutant animals, there seems to be no difference in the 24-hour fear conditioning test, but in the 2 weeks fear conditioning test, there's an increased cue memory, suggesting that the crab E13D mutation, it may be more engaged in the late phase rather than the early phase of the long-term memory. And that is certainly something I want to test in my double mutant animal. And I'm doing the test right now, but I haven't accumulated enough data to make any conclusive conclusion. So I hope I can show you more when I present next year. So for the rigorous and reproducibility, for animal test, I would say the most important thing is we want to make sure that genotype is correct. So all the experimental animals, they were resampled and re-genotyped to make sure correct genotype results, genotype information. And the molecular experiments such as Western and immunostaining, they have or will have they will be repeated to at least three times to make sure the result is valid. And also this may be not very familiar for those who don't do lots of behavior tests, but I will use independent testing assays to confirm each behavioral phenotypes. For example, the repetitive phenotypes, I did the grooming test, and next step I may do some marble bearing to confirm if there's really a repetitive phenotype or some, for example, open field and elevated cross maze to see whether there's a true anxiety phenotype. The results summary. Although the CQO model, the conditional knockout model, is not ideal, it reveals some promising directions. First, the total CRAB level is actually decreased in the MECP2 deficient mice, and that is consistent with our previous finding. Second, in the conditional knockout mice, the CRAB E153D mutation, it can increase the pCRAB versus total CRAB level without causing obvious change in the total CRAB level. And third, increased pCRAB level show little impact on the RTT disease progression and expected lifespan in the CKO mice, but we still need a complete knockout MECP2 model to confirm this result. And finally, the increased pCRAB level may alleviate the repetitive or the anxiety behavior in the MECP2 CKO mice. And I will certainly keep accumulating this data and confirm this finding. So my next step plan is, first I will have to generate a complete knockout line, which I'm doing right now. And we have there are different strategies I can try. And second, I will do more behavior analysis to confirm the reduced compulsiveness or the anxiety phenotype in the double mutant animal. And finally, I will test the behavior. I will do the 2-week serial conditioning assays to explore whether the elevated pCRAB can really improve the late long-term memory in the MECP2 deficit mice. So basically, my rationale of this project is when we talk about disease progression, it's usually we have a disease causal gene, and that will lead to transcriptional dysregulation, cause defects on the cellular level, and that will lead to the behavioral misfunction. But when we try to study this, in my project, I'm doing this reversely. First, I'm looking for any behavioral level change, and when I get a behavioral level change, that will lead us to certain brain regions, and I can focus on certain type of neurons. And after that, I can look for the molecular targets in that brain regions, and I will look for like common targets of CRAB and MECP2, and hopefully and see if that targets this crosstalk between CRAB signaling and MECP2 can have some effects in the Rett syndrome. So finally, I want to give thanks to my lab, Dr. Chang, and all the members in my lab and Tibet's lab who gave us the CRAB mutated mouse models and gave us lots of advice during when I work on the single mutant mouse, and the Jules who give me lots of advice when I did my behavior test, and my committee members. And that's all for my presentation. Thank you all for listening, and I'd like to take questions. That's a good question. So because I don't have lots of animals, so what I did was when I see the mouses becomes like very sick or close to die, I will sacrifice the animal and get the tissue to do the protein analysis. So usually, according to our previous knowledge, the MECP2 expression is stable like after birth. So I would say there shouldn't be much change during their development. But when I get more mice, I will definitely take tissues from different ages and to confirm the protein expression. Well, we do have like PCRs to look at the DNA level. Yeah. Yeah. So I guess something I want to know with all my behavior tests, because we know in the behavior test, and experience what function are mostly affected, and we can expect different techniques. For example, if I really find that if the anxiety phenotype is real in my clinical device, I may look further into the amygdala. And for example, if the long-term memory issue is real, I may look further into the hippocampus, things like that. I see. We can do that. We do have a specific tree in our lab, but currently I think I will mostly focus on the nesting tree, the all neuron knockout, because as we know, in patients, they don't have like specific loss of function in certain cell type. Yeah. I think that is possible, but with this, little circadianism in the so yeah, that's another thing of behavior test. So I would say for behavior test, the most important thing is to be consistent with my testing protocol. So I would do all my behavior tests during the life cycle, and if possible, I will try to do them at the same time of the day. So that So in that way, I can do my best to exclude those possibilities. And also, so one thing that I did with the grooming test is every time I place them in a fresh cage, I'm not recording the grooming behavior in their home cage, because when they are placed in a fresh cage, they are more like active and they're like refreshed, and I guess that can help to exclude some like circadian related issue. Thank you. That's a good point.
No.
I think Ziyao, yeah, may need to stop sharing.
All right, I think we can hear you, Nicole.
Can you hear me?
Oh.
I'm still seeing Ziyao's slide.
Oops.
Oh, that's right. Okay.
All right, well, I'll go ahead in the interest of time, and I'm going to keep this introduction very short, given the complexities and technology and all. It is my honor to introduce our second speaker, Jaime Cordova. Jaime has been in my lab for 4 years, working on the response of E. coli and its close relatives to oxygen availability. Now, if you know Jaime, you know that in addition to his passion for science in general and science outreach, he has particular interest in astrobiology. And I know he must be very excited with the new images coming from Webb Telescope this week. We haven't had a chance to talk about it yet, but wow, let's just say wow. The interest in oxygen availability and it's sort of tied in with astrobiology is one of the things that drew Jaime to our research. And I'm very, very grateful. Not only is he an amazing young scientist, but has arguably the best attitude of anyone I've ever worked with. And I really appreciate your perseverance and resilience and that you approach it all with a smile and a laugh. It makes this whole enterprise of science all the more fun and enjoyable. So today, Jaime is going to talk to us about the response to oxygen availability. And like many of you, we're pretty firmly based in model organism genetics. In this case, our organism is E. coli. And almost everything we know about the response to oxygen availability comes from studying one single strain. But as you'll see from Jaime's presentation today, that grossly underestimates the variability in the genotypes of this organism and the phenotypes. And understanding the connections between the variation between the two is a rather complex process. You may be thinking, well, this sounds like classic population genetics, and in a sense it is, but I remind you that we're working with an organism where not only is it challenging to identify what is a population, but even what is a species. And so Jaime has an extra complex problem here in trying to understand variation in oxygen regulated gene families. And with that, I'll turn it over to Jaime.
Thank you, Nicole. Let me go back. Do you prefer if I use this? Yeah, okay, all right. All
Alrighty, so as Nicole mentioned, the story I'm going to be talking about is trying to look at the variation in the presence and how the presence of bacterial oxygen regulated gene families and how those have evolved. Today, I'm specifically going to be focusing on E. coli, but we do study E. coli and Salmonella, particularly about 170 strains of that. But today, I'm only going to be focusing on about 153, 58 strains of strains of E. coli. So just to give an overview, what I'm going to be talking about is what our goals, I should say, is trying to understand the genomic variation in our E. coli strains, trying to determine the conservation of the oxygen responsive genes in our strains, and then trying to determine how variation is affecting growth in aerobic and anaerobic conditions. And then finally, determining the evolutionary modes of one of those families, particularly the hydrogenases. So just to give a quick overview, going back in time, and Nicole already mentioned my interest in astrobiology. Early Earth, about 2.4 billion years ago, there was little to no oxygen in the atmosphere. It wasn't until cyanobacteria, which were producing enough oxygen in the ocean, and then finally it was released. It escaped, killing off anything that was anaerobic. But basically providing a new challenge for any life that was existing in this formerly anaerobic environment. Well, fast forward to today, where we now have aerobes, which require the presence of oxygen. We have anaerobes, which require the absence of oxygen, and then we have facultative anaerobes, which can survive both in the presence or the absence of oxygen. And our E. coli strains our E. coli and various other bacteria can survive, or are facultative anaerobes, I should say. So the E. coli and Salmonellas are in the family Enterobacteriaceae. They are a large family of gram-negative bacteria. They're made up of both non-pathogenic and pathogenic bacteria. So E. coli obviously get a very bad rep when you hear of, eating bad lettuce at Chipotle or something like that, or tainted meat. But there are some non-pathogenic strains. And again, most of these are facultative anaerobes. So the overall question that we're trying to understand is how much environmentally responsive regulatory variation is present within and between strains of E. coli. And like Nicole mentioned, sort of the motivation behind this is that a lot of the default knowledge for bacterial strains and species, not mostly in the Enterobacteriaceae, comes from the strain, the very awesome model strain, E. coli K12NG1655. So just to go over, what is known about the oxygen responsive subsystems in E. coli K12NG1655 is primarily focused on regulatory systems such as transcription factors, non-coding small RNAs, chemotaxis that are related to chemotaxis motility, metal homeostasis, pH homeostasis and acid stress, and then obviously energy metabolism, nutrient transport, glycolysis, TCA, so on and so forth. So these are the subsystems that are responsive in oxygen, in E. coli K12 NG1655. And the assumption has always been that we can basically see that, or we probably see the same thing in other strains. But what we found so far is the response to different oxygen levels is not necessarily equal amongst all of our strains, which again, we're looking at about 153 strains across the 4 main phylogenetic groups of E. coli. Just to go over, this may look familiar to some of you because I talked about this last year, but basically what we found is that there is similar variation range between E. coli and Salmonella in terms of their growth rates in aerobic and anaerobic conditions. We see a positive correlation between aerobic and anaerobic environments, which basically means that if this particular strain is a good aerobe, meaning it grows well in aerobic conditions, it's usually a good anaerobe also. So it grows well in anaerobic conditions as well. What we've also seen is a high amount of variation in a short period of time for the set of E. colis that are 0157H7s, which are the most pathogenic. They're the ones that get the most bad rep for being pathogenic. And those are these in orange. And we're seeing a high amount of variation in a short amount of time, which they diverge from E. coli K12 only about 5 million years ago. And then we see probably the most important part is that there's a divergence of the K12 strains from the overall trend of the other E. coli strains. So here I have E. coli shown in green, and E. coli K12 are these two in red. So they are kind of okay, aerobes kind of okay anaerobes and kind of mediocre aerobes, which I've since learned and been told, or finally understand, that I can't give E. coli K12 too much of a bad rep because they were designed to not be so strong. So I can't give them too much of a bad rep, but obviously we are seeing that there's a divergence from the other E. coli's. And what we think is that the differences in these, or the difference, what's responsible for these differences in these growth rates, is that they are likely in the systems that are not necessarily directly related to the oxygen responsive systems, but something that's maybe closely related or in the near vicinity of those subsystems. So what we've done since is we've now been looking at the actual we sequenced all of these genomes. We're now able to actually look at the variation that's present in our E. coli strains. What we did is a pangenome analysis, and what we found is that most ortholog groups in our cloud are present in what's called the cloud genome. Again, the goal is to understand the variation in the genetic repertoire of these E. coli strains. We looked at 158 E. coli genomes that covered the 4 main E. coli phylogenetic groups. And we found over 12,200 ortholog groups using an ortho-MCL analysis. And what we found is that about 54% of these ortholog groups are found in that cloud genome, which means that they're only present in about 23 or in less than 23 of our 158 strains. So again, most of these are in the cloud genome. Of the core genome, it's only made up of about 1,442 ortholog groups. And basically what that means is that it's present between 156 to 158 of our strains. And from those 1,442 shared E. coli core genome ortholog groups, about 37% or about 537 of those ortholog groups are metabolism related in our core genome, whether it be inorganic ion transport metabolism or energy production and conversion, things of that sort. I'll also note that about 21% of these from those 1,442 have an unknown function. So it's those are something that would be interesting to study. Now, the next question that we had is obviously, okay, so we've done this pangenome, we've looked at the genetic variation. Where do those oxygen responsive subsystems fall? So who has them, who doesn't? We have, from previous research, there have been about 630, give or take a couple, genes that have been identified as O2 responsive, and about 84% of these are present in most or all of our strains, whether it be in the core genome or what's known as the soft core genome, which is about 150 to 156 of our strains. Now, the question that this brought up is, well, one, what is making up for the response in the strains without some of these genes? So the 79 strains that don't have this, that don't have these in their genome. And more importantly, because a lot of the data comes from E. coli K12 MG1655, this is coming from MG1655. Well, what does that response look like in other strains? Now, that's the next question, which is, how is that affecting growth in aerobic and anaerobic conditions? Ideally, we would be doing RNA-seq for all of these, but I would like to eventually be able to buy a house with that money because it costs roughly $500,000 to be able to sequence all of the, or to do RNA-seq for all of these. So what we're doing is now trying to select a handful of these, which ones are the most interesting, and to really look at how they're responding. But sort of to get to that level, what we've done is we did a pan-GWAS, which allows us to determine that genomic variation and what effect that's having on the O2 response. So just to put it into perspective, or to clarify what a pan-GWAS is that we're doing the genome-wide association study, but instead of looking at all of the genes, we're only looking at the genes that are either in the accessory genome or in the core genome of these strains. Again, trying to correlate those or connect those with the growth rates in the aerobic conditions and the anaerobic conditions. And this is not meant for you all to be able to actually be able to decipher what this is, but basically what I'm trying to show here, so this is in aerobic conditions. These are 153 strains that we're looking at here. And what we found using that pan-GWAS analysis is we identified about 241 genes that seem to be associated with the growth rate in aerobic conditions. Of particular interest are the strains that are in this in this area of the phylogenetic tree of our E. coli strains. Basically, what I'm curious of is, well, one, let's look into more detail what those growth rates are for these particular strains, but more importantly, what are these guys doing that these strains don't have? And if we look at the anaerobic conditions, and really prepare yourself for this one, we see about 755 genes that are associated with growth rate in anaerobic conditions. And again, we see in the same area of the phylogeny, we see very similar patterns where there are these genes that are present in that particular group, but not the rest of our E. coli strains. And I'll note that while I don't necessarily show it up here, and looking at what these genes that have been identified, they're not really necessarily connected, or they're not necessarily in that list of those that have been identified in the past, which sort of goes back to what we had hypothesized, that these changes in the growth rate, or these differences in the growth rates, may not necessarily be strictly in the O2 related subsystems. So as I mentioned, with some of these genes being subphylogeny specific, we're hoping to use this data to select a subset of our 182 strains for further analysis, including RNA-seq. And the last part that I wanted to talk about is how have oxygen responsive genes evolved in our strains. We're particularly focusing on the hydrogenases here. And basically what the goal is trying to detect duplication or horizontal gene transfer events that are impacting how the oxygen availability response varies. So the way that this works is we're looking for trends, again, in the evolutionary processes. Are there duplications? Are they evolving through duplications, through horizontal gene transfers, or simply speciations? The way that we do this is we take a gene tree, which gives us the the history of how the gene has evolved. We take a species tree, which tells us the history of how the species has evolved, how the species has evolved, and we reconcile those, which what that allows us to do is, again, tell whether we're seeing a duplication, transfer, speciation, or gene loss. And the reason why we're interested in this is because there are known trends in the evolutionary processes based on oxygen responsiveness and gene function. So for example, virulence and metabolism have been shown in some, at certain phylogenetic levels to be evolving through horizontal gene transfer. So we're focusing on the hydrogenases. It's very difficult to show stuff when you're working with 160 strains. So again, this is, so the goal of this is just simply to show you, you're not meant to look at the actual labels, but I'll explain what we're looking at here. So we're focusing on the hydrogenases, and what the hydrogenases are responsible for is they catalyze a reversible reaction that's responsible for converting protons to dihydrogen. We know that from previous work in the lab that there is a transcriptional variation in other strains that are E. coli strains or closely related in response to oxygen. And many of these, the enterobacteria, so not just limited to E. coli and Salmonella, encode multiple hydrogenase systems. For example, E. coli K12 MG1655 encodes 4 out of 5. In our case, it may be a little bit difficult to see. The majority of our strains encode 2 of the hydrogenase systems that we're looking at, with only 1 or 2 encoding 4, and the rest encoding 3. And I will point out that there is 2 that I encode 1 out here, but those are our outgroups, just in case that comes up. So what we found is that the way that these hydrogenases have evolved is is primarily through speciations. And this, in a way, sort of makes sense because these are all E. coli strains. So it's possible that because we're looking at such a focused phylogenetic level that they primarily, there hasn't been much transfer. As opposed to a prior analysis that we did using 110 enterobacteriaceae. So now we're not just looking at E. coli and Salmonellas, but we're looking at a kind of zoomed out phylogenetic level. We found that they primarily evolved through horizontal gene transfers. Again, this is likely due to a more focused taxonomic level. Now, so in terms of rigor and reproducibility, the goal, or what we do, or what I do, is maintaining meticulous notes of the process of what we're doing. So what software version am I using? Where did the software come from? if I'm communicating with the authors of this software, because I have to do that a lot to double check things, keeping those emails where the scripts originate from, things like that. So we are keeping meticulous notes on how we do this, and even sometimes running these processes a couple of times to make sure that they agree with each other. So to conclude, most of our E. coli ortholog groups are part of the cloud genome, which again means that they're present in less than 23 of our strains. Despite their relatedness, 84% of the E. coli K12 MG1655 oxygen responsive genes are found in most of our strains, but the question is what's making up for that in the rest, so the rest of the 16%. We identified some lineage-specific genes that appear to be associated with the growth rates. And at our E. coli and Salmonella-focused phylogeny level, the hydrogenases appear to have evolved through speciation, as opposed to on a much more zoomed-out level, where they've appeared to have evolved through horizontal gene transfer. And I'd like to thank my lab, including former members Dave Assetti and Drew Hazley, GSTP for my funding, my committee, and then the parental lab heads. So, and I'll take any questions.
I have one question.
Okay. Don't make them too hard, Chris.
It's like that. So for the phylogeny, is that from the Colloquium?
The phylogeny for all of our strains?
Yeah.
No. So what we did there is we took we did the ortho MCL analysis. Well, I guess you could say it is from the core genome. We did the ortho MCL analysis. We took all of the strains that only showed, or we took all of the ortholog groups that only showed one copy, and solely one copy in each of our strains, concatenated those, and that produced our phylogeny. Now, and just for the sake of mentioning, I know that there is, I don't know who's here is a hardcore phylogeneticist. I know that there's the argument of doing concatenation versus doing other methods. Well, we also did the more, I guess you could say, more stringent methods that are much more computationally expensive, and we found the same species tree.
So it's a subset of the protein.
Correct.
Is there gene species tree discordance?
Gene species tree discordance.
Even within the subset that you use?
Sorry, remind me what discordance means.
So just like if you do an individual tree from a sample gene from the set that you actually did, do you know if they tend to match the species tree?
Oh, okay. No. So far with the ones that we've looked at, because so far we've done this with the oxygen responsive. Oh, so far we've done this with the oxygen responsive strains and just a subset of those. Well, we did run into some of that discordance, and we're trying to work out how to do that. We're using we're using a piece of software that's associated with another piece of software called Ranger GTL, which is how we're doing these reconciliations that basically help us figure out or solve that discordance. And that's primarily been shown as polytomies, or that's mostly how that discordance is appearing. But we're basically helping these gene trees using an algorithm that also uses a species tree to help basically solve those polytomies.
So it's a very effective.
And the ones that we've seen so far, I don't know. I wouldn't be able to give you a percentage or say if it's high or low, but I can say that basically one of the issues that we've run into is that because these are so closely related, some of these sequences tend to be almost identical. So that's what we're trying to solve right now, which so far it seems like the algorithm that we're using that I mentioned uses a species tree to help guide solving those polytomies. Seems to be working, but we're trying to look at other things to see if just to confirm it.
Yeah. And also since it's like, I mean, if you're looking at the copy of well, they're probably also higher conservation. So.
Yeah.
Interesting. Other question. You had in the looking at the gene presence with the oxygen and growth response. genes. Do you have any like ecological data about those blades in the top or in the middle that would give you any more information about what they do?
Yes, so we have some limited data in terms of where they came from. Not for all of these though, but the interesting thing is that a lot of these have been cataloged pretty well. So the data is out there. We just haven't gotten to that step yet, but that is one of our next steps.
Nothing obvious.
Nothing obvious. Yeah.
I have a related question.
Yeah.
I was interested in what types, what are you, do they fall into specific geo categories? Are they mostly function or what kinds of genes?
Yeah, and sorry that I realized I didn't list that. So from, I haven't taken a specific like detailed look at it, but just in kind of looking through the data, it seems that a lot of them are proteins of unknown function. That's what it seems like so far. However, I will note that the way that we're putting these into the or categorizing them, I guess, with the geo labels, we're using a particular program that takes from various sources, but it may not. What I realized recently is it may not be the most updated. So there may be new things or new identifications that we just haven't found yet, and that's one of my next steps is to, okay, is this really a known function or has it been identified since, maybe even in something closely related, if not E. coli?
I have a related question.
Yeah.
Because there is these protein family called Gauge oxidases that were really recently identified in bacteria because people only believed that they were presenting eukaryotes, but something like maybe less than 5 years ago, they showed that there was in bacteria. And they are really interesting because as far as we know, they sense oxygen. As their primary function is sense oxygen and use it to produce free radicals for many different functions in eukaryotes. So they are not, you're not going to find many isoforms of these proteins, probably only one. So I know that you're looking for you have gigantic data of proteins,
Yeah.
but maybe, this is one example of these unknown functional protein because it was just recent. But anyway, I'm just suggesting you to take a look at this because to me it sounds kind of obvious to look at this protein.
Yeah, no, definitely.
Because of these primary functions.
Yeah, absolutely. And sorry, what was the gene?
pH oxidase.
Okay, all right.
A lot of people are interested in them. So
Okay.
NADPH. Oh yeah, NADPH.
Okay, all right, I'll remember that.
Yeah, so I think it's super related to your question.
Yeah, I'll definitely look into that. Thank you. Or was there another question?
I was just curious
Yeah.
To what extent you are focusing on protein coding genes and whether you have any plans to look into non-coding RNAs.
Yeah, so far this is all in protein coding genes, but that is also one of our next steps is to look at the at non-protein coding. Basically, the rate limiting factor is always been that the software doesn't always work straight out of the box, and I'm sure any bioinformatician can tell you that. But that is the next step.
Sure, it seems like you might have some really cool opportunities, like leveraging the system that you have with having some so closely related species and having like a well-resolved phylogenetic tree that you might be able to, like, as you were pointing out in the beginning, identify a couple of species that are as closely related as possible phylogenetically, but have the greatest variation in terms of response, and then like really take a deep dive, and that might be a good place to look for maybe some more nuanced differences or more recent evolutionary events like the
Yeah.
conflict.
Yeah. No, it's a great idea. Thank you. Yeah.
So I was just thinking about, K-12 as being our anchor for everything. It's kind of not really a great anchor, right? So you showed in your plot early on that it's kind of poor at growing both in oxygen and without oxygen. And I think that's probably because it has a lot of mutations that are acquired in the lab setting, like at least factor 2 was mutated, the purine metabolism gene was mutated, right? So I would encourage you, when you get your new data sets, to think beyond that as the anchor, because I think that it could be really confusing, and that there has been some evidence recently of people who are doing like comparative genomics using gene phenotypes that have seen where K12 is basically an outlier amongst other coli types. They weren't looking at anaerobic growth, they did all their experiments aerobically, but they definitely found K12 as the outlier.
Okay. Yeah, good to keep in mind. Any other questions? Are there any questions on the chat? All right. Cool. Thank you, everyone.